Hello.
Hi, Beth, you okay?
Yeah, good. Yes. Jorge's talking at 1:15.
Cool.
So, yeah.
Hi, Beth. Can you hear me?
Yes, I can hear you.
Cool.
Is the sound all right on mine? Can you
Yes,
hear me? Okay, good.
I've just had to turn it down actually. Going to turn it back up a bit.
Oh, maybe I'll stop shouting.
Ooh. Right, I'm ready. This is always that moment when no one's going to turn up.
Yeah.
We've had a fair bit of interest in it, so I hope that's not the case.
They usually start dribbling in about like 1 minute, 2, don't they?
Have we got an advert up on Facebook?
We've got like 100 adverts up on Facebook and Twitter.
That many?
No, it's we've got it on the Facebook page and we've got it in the group. So there's no excuse for, and it's gone out on Hospify.
Okay. So we have Jorge till about what, 45 minutes, something like that.
Yeah, something like that, maybe half an hour. I don't think it's a topic where the patients are necessarily going to have a lot of questions to discuss.
What is it?
Persulfidation.
Oh,
So it's fairly molecular.
and the relevance of that is
Did you watch the video?
no.
Maybe I'll start with that. It's a post-translational modification
Yeah.
that Aspergillus and humans both use to react as quickly as possible. So when the Aspergillus is trying to set up an infection and when the lungs are trying to prevent the infection, they both persulfidate a lot of their proteins. And if you take that mechanism out, it has a big effect. And it might be druggable.
But is it, if it's druggable, is the drug likely to be toxic if the human side is also using it?
I think we don't know enough about it's a really not very well known one. It's not like acetylation we've got loads of stuff for, but I think it's a quite a new one.
I've not heard him talk about this before, so that should be interesting.
It's got a lot of pretty pictures,
Well, can't
and
be
he's a
pretty.
very enthusiastic, nice guy, and he didn't cancel. So
Malcolm used to love his pretty pictures. So let's keep it up.
yeah.
Come in. Gwyneth's doing the dishes.
Yeah, sorry, yes, I am. I'm just muting myself. I'm just getting lunch. Sorry.
Lunch, don't make me feel hungry.
Sorry?
I'm feeling hungry now.
I'm sorry.
You
What, yeah?
never eat dinner. You never eat dinner before these. I always have to have dinner before we start.
Yeah, I just can't fit it in with the calcium tablets and the 4 hours, 2 hours.
How to turn the thing.
Having something nice to eat.
I'm
Sandwiches.
having what I call Spanish chicken, which
Ooh.
is an adapted recipe with a load of veg, normal stuff.
And chorizo.
Sorry?
Chorizo.
Oh no, I can't have chorizo. No,
Poor thing.
I know my diet's really bland.
It is very bad for you, obviously. Processed red meat. Morning, Doug and Christy. No, sorry, not morning. Afternoon.
Afternoon.
Hello.
Have you all
You all
been?
right?
We're
Oh, yeah.
okay. We're still swerving to COVID. It's pretty good.
Yeah, the hospitalization rate's not going up anywhere near like last time.
No, I've just been looking at the figures.
Yeah, it does look bad when you look at cases, but
Yeah,
if it's all
except that the rate of cases seems to be leveling out. Well,
Mm.
the gov.uk figures are going up steeply. I don't think the leveling out has fed into those figures yet.
It's the schools that are the problem, isn't it?
Seems to be.
So many children being sent home.
Did
Yeah.
you see I didn't read the full article, but they reckon some kids were using lemon juice
Yeah.
to do a fake positive test and get everyone sent home. Little swines.
That'd be something my youngest would do when she's older.
You can just guarantee as soon as people get hold of anything, they'll find a way of
Mm.
Dead now.
doing something.
I don't think it helps for those that perhaps, mind you, if you're going to do it, you probably will know. But you know, for those that didn't know, who now think, oh, lemon juice.
Yeah,
It's a nice day. We'll have it off. You know, have a day off.
10 days off.
They have to get a PCR to back it up then, don't they?
Only the ones who are frozen.
Yes, you do.
And come back.
And I
We
think
froze for a minute then.
I think you're just working from home rather than getting a day off. With this, well, certainly he was with my son's school. He had to carry on, but he's got not an exam today, but sort of an internal exam. He's got to do his physics today. So we talked about things like forces. What was he particularly going on about? I like to talk about ladders, leaning against the wall, which is a good old classic.
Yeah,
which
triangle.
I never understood properly when I was doing my A levels. He was talking about something else. Can't remember. At least he's aware of I think he knows what physics is, but that can be.
How old is he?
He's 14, so just about to enter his O-level years, GCSE years. What are they called now? Years? Don't know.
Still GCSEs at the moment, aren't they? They're just all numbers instead of.
Do they
letters.
still have to learn laws like Ohm's law and
Yeah,
Newton's laws?
those are the basics.
Yes, but do they have to learn them as, you know, like we did? I mean, these days they don't tend to learn by rote, do they?
I noticed somewhere, I don't know how long ago it was, you actually got provided with some of these equations when you were, which would have worked for me because I could never remember an equation. But, managed to pass just about. Is it everybody on holiday, do you think?
Oh, watching the tennis, I bet.
I'm on
Yeah.
today.
We had 11 people register interest on Facebook, so why? I don't know. Maybe, yeah, tennis.
Did you actually send the number out that you put into Zoom?
Yeah.
I used an old when we used to get texts. I always pick it up from there.
Yeah, it's all there in the invite.
At least the members of staff aren't outnumbering
Almost.
yet. But the all-time low that we've ever had was 2, and I'm sure that was in July.
It's like seeing anything at the Edinburgh Festival. There's more people on stage than in the audience, and it's just really embarrassing.
Well, I don't understand it because no 13 people responded to the event. I don't. I'm confused.
Well, we're going to have a debate about this, aren't we?
Yeah. I mean, maybe some of them are watching the streaming.
Do,
I don't know how we can count.
That's true.
the big question is, are we going to return to coming back to the hospital,
Ah, well.
or whether we're going to carry on online?
Would you like to return to coming to the hospital? Is it a trip out?
Well, we haven't met anybody for 2 years.
Well, it'll be a bit overwhelming then. You don't need to do that, do you? I think probably we should offer the option to people that, but we would also we're trying to do a hybrid thing where they're both happening at the same time, which we tried to do a few times before, but now everyone's a lot more familiar with Zoom, so that might work a bit better.
I suppose in the winter, if you get bouts of really bad weather, then being on Zoom is really handy, isn't it?
This is it. Clearly, the weather's just too nice. People are out just doing stuff.
Living their lives.
Yeah. Or we
You're
could
trying to
use
say we haven't got any.
Well, you just said you've not met anyone.
No, we haven't. No lives.
Yeah, we could do. We can maybe even do sort of more than one type of thing. So we could have things that are more sort of like anything a bit sort of sciencey or just information. We could do that more on Zoom, and then have the discussions as the in-person bit, or have people bring their crafts along, or the things where we grab other members of. Sorry, my words aren't very good today. Like if we've got Phil helping us with something, I think that would work better in person. Maybe.
Excuse me, the phone's going.
No, you can't go.
Oh,
Now we do outnumber them.
Yeah, I suppose a lot of things. I mean, are you proposing a red button channel there, Beth? So we have another channel working in parallel to this one.
I was thinking more like, so like at the moment, there's segments out of the meetings that get snipped out and then individually put on YouTube.
Yeah.
So I think those sorts of things could be done on Zoom if people, like if the weather's really bad and people don't want to come in. But then
Yeah.
like all the chatting and doing the exercises and stuff, we probably want to be together.
Yeah.
Do you think it might put people off the fact that it's being recorded?
I've not heard anyone say that, but then people might be shy to say that. So
Well, for
No.
the first time today, as I logged on, it actually said this is being recorded, and it hasn't done that before. I'm just wondering whether people going, ooh,
Yeah,
maybe.
maybe.
oh, it might be worth asking the question on Facebook.
Yes. So
Yeah.
well, we take trouble to blur everyone's faces out. Like this, if we're recording, if this ends up on tape, then on my computer, the top line is us, so we'll all get to stay. On the bottom line is you guys, and you'll all be blurred out, so no one can tell who you are. Because that's regulations, we have to do that.
Mm.
Yeah, some discussion bits you just have to cut out completely because like people are saying their names as well.
Mm.
Yes, that's true. There's only so much we can do, isn't there?
Yeah.
And we try to keep it private. We don't make the recordings widely available. I don't think.
No, I think we've got like 5 followers on YouTube now.
Yeah. One of them
This-
could be Donald Trump.
There's been a lot of talk and a lot of thing on the news about the fungus in India.
Yeah.
The mucormycosis, yeah,
Mm.
horrific.
But that's not Aspergillosis, is it?
No, it's quite a close relative of it. So like the big Aspergillus conference that happens every 2 years, that's now Aspergillus and mucormycosis. So they're quite similar diseases, but mucormycosis is a lot worse and really goes for people that have got diabetes, unfortunately.
So is it an invasive fungus?
Yeah. And especially sort of around the head and face. So there's been a lot of people like losing, they have to have their eyeball removed or something. And
is that because
mortality
of
rate.
the oxygen mask?
I don't think so, no. That's just a feature of mucormycosis generally, and that's just the bit that it preferentially. And it does infect lungs as well quite often. So do you remember in the news last year there was a girl in Stockport died of mucormycosis. She'd caught it at stables.
All right.
They didn't know she was diabetic because she had no symptoms, and the first thing they knew was she just had massive hemoptysis and didn't make it, and all the A&E staff had to have counselling because it was just completely it seemed like out of nowhere, but it's so like that was a huge news story just because, you know, just one case of it because it's so rare here. In India, they already were having 70 times the number of cases than they do in other countries. And it's, and there's a high rate of diabetes that isn't always very well controlled there. So. And
What was the fungal infection they got from the hospital with the pigeons?
That would probably be cryptococcus.
Crypto.
Which country was it?
Here.
Oh, in this
Yeah, it was here.
Oh, I don't
The,
know.
was it a fairly new hospital, new wing of a hospital, and the air conditioner was taking in pigeon droppings or something like that. I mean, it's a while ago.
It was in the Queen Elizabeth in Glasgow. It was cryptococcus.
Oh, yeah. Trouble is, so many different fungi can live in bird droppings, bat droppings. There's lots of different ones, like different parts of the world will have, it'll be a different fungus, but it'll do the same job.
Yeah.
And it's especially in places where maybe people are doing construction work and they don't have such strong regulations about PPE. You can get very big outbreaks, unfortunately. Hello, is that Jorge? Are you on the call now? Oh, hi.
Hello. Yes.
Hello.
Yeah, I'm already.
Morning. Cool. So this is Jorge Amich. He's our guest speaker for today. So he had a paper out recently that is quite exciting, and he's going to tell us about that today. There's only a few people on the call. We're streaming it and recording it as well. So it looks like you're talking to 3 people, but actually it's going to be more. So
I would be happy to talk to a few people as well. So people who are here in any format that are interested, it's my pleasure to talk to you all. So let me share my screen. Okay, is that fine? Can you all see it?
Yes, it's fine.
Okay, so hello everybody. Good afternoon. It's a real pleasure to be here with you and talk a little bit about the research that we are doing in the lab. So already gave a seminar to you, I think like 2 or 3 years ago already, so quite a long time. So with all the COVID situation, it's been difficult for everybody, and it's really a pity that we cannot meet in person. It's always much more fun, but still, it's a great pleasure to present to you this work and see that you know what we are doing in the university, and our hope is that in the future all this work can help you, can help to fight this terrible disease. So. Well, this is the title of the paper that we just published. It just came out last month, so it looks complex, doesn't it? Fungal and host protein presulfidation are functionally correlated and modulate both virulence and antifungal response. So that's the title. I'm going to explain to you what it means and why this is relevant. So the first thing I want to say is that this is about a protein modification. So it's a protein post-translational modification. And now here I'm going back to your probably biology studies back at school or university. And I hope that you are slightly familiar with this. This is called the central dogma of biology, which basically says that all our genetic information is contained in DNA, which can self-replicate, of course. So that's how it's transferred from cell to cell when cells divide. And they'll DNA, and let me, should be able to use this so you can see. So DNA is transcribed into RNA, which is like a messenger molecule that is like reading the information from cDNA, so it can be transformed or translated into proteins, which are the actual functional components that execute the functions in the cell, right? So this is the very basics, but of course, this is getting more and more complicated as research advances, so we understand the processes a little bit more. So actually, this RNA is just one subtype of the various subtypes of RNA which exist, and it's called messenger RNA because it has the information, so it's a message of the information to be translated into proteins. And when this molecule is synthesized, it has some pre-forms that can be further modulated, so there are more controlled steps in between. As I said, this is only one of the subtypes of RNA molecules. We have several others like tRNA, transferase RNA, or rRNA, ribosomic RNA, and these directly are involved in the translation process itself, so converting this information into proteins. There are also other types of RNA called siRNA, small interference RNA, which can control the mRNA molecules after they have been produced. You see, this is getting more and more complicated. But then what my investigation has focused on is in the last step of the modifications that can be added to these molecules, and are modifications that can be added to proteins. So once proteins has been synthesized, they exert the functions. But on these molecules, several modifications can occur as well. Some of them are very well known, as I guess you have heard of them. One of them is phosphorylation, so adding phosphate groups to different residues in the protein molecule. There's many others. This one also quite well known called methylation. And the one we are working with is called persulfidation. So we are focusing on this particular modification of proteins and how this is important for the infection process. So now you may ask, so why could this modification, why could in general be all these modification important for the infection process? So I always like to pose this analogy, and that infections are like a battle between the attackers, the pathogens, and us, the defenders, trying to defend the castle, which is the analogy of our body. Of course, during each battle, each contender needs to adapt to the other's strategy. So depending on what weapons the attacker deploys, the defender needs to deploy different types of strategies. So transcriptional adaptation is actively studied and known to be very, very relevant. And this, we come back to this stage, refers to this step, how DNA forms RNA, which in the end will be the precursors of the proteins, right? So this is recognized to be a very important step in adaptation on both sides, from the pathogen and the host, and is actively studied. But this refers to the construction of proteins again, so the construction of blocks. So imagine that each protein could be a different weapon, tower in battering ram, a siege tower, or a catapult. However, the post-translational modifications, which are added after the translational process to the proteins, are very fast-acting mechanisms, and therefore they enable a very quick adaptation. So now think about a pathogen who has created a strategy of constructing a battering ram, for example. But then the defenders start pouring boiling oil on top of it. So what are the attackers going to do? They're just going to abandon this strategy and mount a completely different one? Or could they just add a small modification to this protein? So this protein is still active and effective, and they can continue using this strategy. So this is the idea of investigating the importance of post-translational modifications for both pathogen proteins and host proteins. Okay, so the one we are interested in, as I mentioned before, is called persulfidation, so has been called in the past sulfhydration. Basically, what this modification involves is the attachment of an extra sulfide group to the thiol groups of the cysteine amino acids of certain proteins. So this group, which is normally just one S and is called thiol group, is modified and now it contains 2S, and this is a persulfide group. So this small modification is known that can increase or decrease the specific activity of certain proteins. And it's been quite actively studied in humans, in mammalian cells, and it's known to have a prominent regulatory role in various physiological functions. It's also known to be involved in inflammation, but its role in the actual innate or immune response, it wasn't very clear. So we decided to check investigate if this might be important for both the pathogen, Aspergillosis fumigatus, and also the host defense. So let's first talk about the pathogen. So what we did is construct mutant strains, and I guess you already know what that is. It's just we eliminate specific genes in the fungal genome. So we constructed 3 independent mutants, 1, 2, 3, and each of these strains now is being deleted in one gene, cannot produce one of the proteins that have been described to be important to perform persulfidation in other cells. So the proteins that actually incorporate S into proteins, right? So there are 3 of them described, and we have deleted all 3 independently. And of course, what we have done is to measure the level of this modification of persulfidation in the total proteins of the fungus. So to do that, we have used a protocol that has been optimized by our collaborator, Filipovic, who is based in Germany. Basically, what this protocol involves is labeling all proteins in the cell with a green fluorophore, so we can see all the proteins here, and then specifically the proteins that have been subjected to the persulfidation modification in red here. So we can see how much is persulfidating and normalize the values to the total protein content to account for small differences that could happen in the loading. So we measure that in the whole protein extract, which we can see here, loaded on a gel in the lab, and then also on whole hyphae using microscopy in this other part. So we can apply the protocol slightly modified in both fractions. And when we measure the level of persulfidation in the wild type hyphae, so the hyphae that have all its genes and single specific mutants, you can see here in these quantifications that particularly this mutant that is deleted in this gene here has a significantly lower level of persulfidation. So inside this cell, the proteins have less amount of modification than in a normal cell. We try to construct clone mutant, so having a strain, fumigated strains, which is deleted in more than one gene at the same time. And the idea is that probably we wanted to decrease this level even further to go down because we still have around 50% here, right? So we wanted to go down, but it seems that this cannot be done. So what we propose is that this post-translational modification is essential for life, so it cannot be completely destroyed. We can, tamper with it a little bit and decrease its level, but we cannot completely abrogate it because then the cells just cannot survive. So all the analyses that we are doing are actually when we decrease the levels of this modification a little bit, not when we eliminate them. So we've made quite a lot of analysis with this mutant strain to understand what's happening and how it works, but I'm just going to show you the actual functional consequence. And this is that when we challenge the spores of Aspergillus fumigatus with macrophages, and these are macrophages coming from mice and a cell line from human macrophages, in both cases, when we challenge the spores with macrophages, you can see that the number, the percentage of mutant conidia that are killed is higher than the percentage of wild type conidia. So that means that the mutant has a lower capacity to survive in the presence of macrophages. We went on and assayed mouse infection experiments that we need to do as a last stage, always to confirm the validity of our results. So we actually have 2 different models. One is called neocopennic, and this model mimics to some extent the situation in neutropenic patients. So for example, patients that have been subjected to hematopoietic stem cell transplantation. And then we also have a corticosteroid model, which mimics to some extent patients who are under corticosteroid treatment, for example, patients with GVHD, or nowadays patients with COVID are subjected to this treatment as well. So these mice are immunosuppressed, same as patients under these treatments. And when we are infected with Aspergillus, they normally die of infection. So we have, when they're infected with wild type conidia, or wild type Aspergillus, they die of infection quite significantly. But when we infect them with a mutant strain, remember that has lower levels of persulfidation, there is a higher survival rate. So leukopenic, there is a significant decrease here, and it's even broader in the corticosteroid model. So basically, as a summary of the effect on the pathogen, this means that if we impair with the post-translational modification, so this modification of the proteins, we reduce the virulence capacity of Aspergillus fumigatus. All right? This on the host side. On the pathogen side. Now we go to the host side. So the idea is a little bit the same, but before getting into all the analysis, which is quite complex with host cells, mammalian cells, we wanted to confirm that this was actually going to be important. So to do that, we team up with Agostinho Carvalho, who's based in Portugal, and he has access to genome databases of patients that have undergone this hematopoietic stem cell transplantation, and they have been followed up, and we know which patients developed invasive pulmonary aspergillosis and which patients did not. So he can do associations to see if the presence of specific variations in the human genome associate with a higher risk of suffering invasive pulmonary aspergillosis. So he found that in the human gene that accounts that exert this post-translational modification, which is called CTH, there is a particular nucleotide change that also causes an amino acid change in the protein. So there is one amino acid in the protein which is different in this population. We expressed this protein recombinantly in the lab, and we got the wild type or the normal version of the protein, and then this other version of the protein with this small mutation. And we confirm using an enzymatic activity in the lab that the protein with this small modification is slightly less active than the normal protein. So this means that this modification has an actual functional consequence. Okay, and then since this was promising, our team ran this association study that I mentioned. And what we found is that when this modification was present, is this one, in the recipients, summing up, there was a higher risk of suffering invasive pulmonary aspergillosis. So this really associates, this presence of this SNP in the protein that exerts persulfidation associates with a higher risk of aspergillosis. But it was quite surprising that it's in the recipient compartment. So, sorry, in hematopoietic stem cell transplant, I guess you all know how it works. A patient is completely immunosuppressed, so its immune system is destroyed, normally because there is a malignancy, like a leukemia or similar. And then, so the immune system is completely destroyed, and new immune system, new stem cells from a different patient, a donor, is transferred to reconstitute a healthy immune system, right? So the analysis checks the genotypes of both the recipient and the donor, because they are going to be different. So it was surprising we didn't see an association in the donor. So the immune system, the cells that create the immune system, did not really matter if they have this SNP or not. But the cells of the host, of the recipient, did. So other cells, not those that actually create the immune system, seem to be important, that have correct persulfidation levels. So of course, we were very intrigued, and we wanted to understand why a little bit better. So for that, we contacted Andreas Weihach, also based in Germany. And in his lab, they host a mouse line, which is a knockout of this particular gene. So these mice do not have the gene that exerts persulfidation. And it is known, and it's been studied, that they do have lower levels of persulfidation in the lungs, the site of Aspergillus infection, of course. So what we did is we took neutrophils from these mice, which are of course immune cells, and then alveolar macrophages, which are cells that are located inside the alveoli, inside the lungs. And these cells, even if they are immune cells, because they are macrophages, they survive for very long periods of time. So even in hematopoietic stem cell transplant recipients, they have their own alveolar macrophages for very long periods of time. So these are cells coming from the donor, and these are cells, at least for some time, coming from the recipient. So we compare what happens when we challenge each of them with Aspergillus. We first challenge neutrophils. And as you can see, same as we observed before with macrophages in the figure that I showed you, right? So the mutant Aspergillus is killed at a higher rate than the wild type Aspergillus. Again, showing that persulfidation in the fungus is important. But there was no difference whether the neutrophils were coming from the normal mice or the mutant mice. They were able to kill Aspergillus at the same rate. Surprise came in arbor macrophages. Now here you can see how the macrophages coming from the mutant mice had a very significant impairment in the capacity to kill Aspergillus. So correct levels of this post-translational modification of persulfidation are important for arbor macrophages to be able to kill Aspergillus at a normal rate. We wanted to further confirm this, so now we used a human cell line. So this is an immortalized cell line that we can grow over and over in the lab. And it's an alveolar epithelial cell line. So this also reflects the recipient part of the transplant. So these are the cells that are in your epithelium inside your lungs. So when there is an hematopoietic stem cell transplant, you would still have your own cells. So we got this cell line and we constructed a mutant using this novel CRISPR-Cas9 technology. And what you can see here is that the normal cells express the protein, this CTH protein, but the mutant cells that we constructed do not express this protein. So we measured, again, the levels of persulfidation in this cell line that we have constructed. And the same as we observed before with Aspergillus mutant, the level of persulfidation is significantly decreased in this cell line. It's not completely abrogated. So again, we didn't even try to construct a double mutant because no one has been able to construct it. So we propose that this modification is essential for life. It cannot be completely abrogated. But again, we have a cell line that produces significantly less levels of the modification in the proteins. And once again, now we challenge these cells with Aspergillus conidia, and we observe exactly the same as with alveolar macrophages. So the mutant Aspergillus is killed at a higher level, so persulfidation is important for the fungus, but then also the mutant cell line could kill Aspergillus at a lower rate than the normal cells. So again, this modification is important for the host cells to be able to kill the pathogen. Not only we look at direct killing, we looked at several other readouts of protein function, and one of them that was very significant and important is the production of cytokines, which are these molecules that help to orchestrate a proper immune response against pathogens. So one of them, which is very important in epithelial cells, was completely dysregulated in the mutant cell line. So it seems that not only the cells are not able to kill the pathogen right, but they also are not able to orchestrate a proper immune response. And we believe that's why people with impairment in this protein are at higher risk of suffering infection. Okay, then if you remember the complex title we had at the beginning, there was this worry interdependency. So this is going to be brief, but we just wonder, okay, so if the levels of persulfidation in the host correlate with their capacity of killing the fungus, and on the other hand, the level of persulfidation in the fungus correlate with its capacity to survive the attack by the host, does it mean that they are balanced out? So the higher the levels of one, the higher the levels of the other. So we measured them in parallel using several methods. And here is one of them. We grew Aspergillus on the cells, and we grew wild type Aspergillus and mutant Aspergillus on the normal cells and the mutant cells. And as I explained to you before, we measured the levels of persulfidation inside the cells, the fungal cells. And what we found is that, as we were hypothesizing, the levels of persulfidation in the fungal cells are highest when the wild type Aspergillus is challenged with wild type normal cells. But when the wild type Aspergillus is challenged with the mutant cells, which have lower levels of persulfidation, the level of persulfidation that the fungus needs, to counteract the attack of the host is lower. So they are balanced out. Why is that important? Because we think we can exploit this for treatment. So the last this is going to connect now with the future work that we're planning to do, is that we have observed that the level of persulfidation in the mutant increases with stress, like, for example, the challenge in the fungus with the cells is a stress for the fungus, so it increases the levels of persulfidation. If we challenge with other stress, oxidative stress, for example, we also see an increase of persulfidation levels in the wild-type Aspergillus. But when we add a sulfite donor, we do not see this increment. Why is that important? Because when we use a sulfite donor, and this is not our work, this is by our collaborator here, when we use a sulfite donor, for example, this one here, the levels of persulfidation increase very significantly in the host cells. So this is the basal level. See, the red signal is quite faint here. When we use a sulfite donor, it becomes much more intense. So sulfite donors seem to be able to increase the persulfidation in the host cells, but not in the fungal cells. If you remember, as I just told you, the levels of persulfidation in the host cells correlate with the capacity to kill in the fungus. So if we can increase the levels in the host cells, we will kill the fungus much better. But we need to make sure that we are not increasing the persulfidation in the host, in the fungus, because otherwise the fungus would be able to defend itself, right? So we've made one experiment so far to test that, and we'll hope to keep going. So we did exactly that. So we treated the cells with a sulfite donor and really corroborated that if we add a sulfite donor, the normal cells are able to kill Aspergillus much more than when there is no sulfite donor. But this, with all these controls, also means that the sulfite donor is able to increase the killing capacity of the host, but is not able to increase the deficient, so the capacity of survival for the host. So what we are aiming to do is to develop a treatment to increase persulfidation in the lung cells, so they can increase their capacity to kill the fungus. But we believe that will not affect the fungus, so the fungus will still be as sensitive as normal to the killing capacity of the cells. So that's it. The plans for future work are, well, understand a little bit better why the host cells are able to kill the fungus better. Then determine if this treatment that increases host persulfidation in the lungs has a beneficial effect on infection, and we will try both on its own, just that treatment, and in combination with the existing antifungal drugs to see if there is a synergistic effect. And then I didn't show you anything about this, but we also would like to test if a recently developed inhibitor of the bacterial, but not the human enzyme, can also be used in Aspergillus fumigatus infection. So that could be a potential antifungal treatment. So now the idea would be at the same time to increase host persulfidation and decrease fungal persulfidation. So we make this gap before the killing capacity of the host and the defending capacity of the fungus even bigger. And hopefully we achieve a better defense in the lungs. All right, so just like to thank all the people that have participated in this work. This girl, Monica, was the one that did most of the experiments in the lab. And here is everybody of the MFIC, Manchester Fungal Infection Group. So this is mostly the lab side, the university side of the fungal group. I think you can all know David was here with us. Most of you know Paul as well, and probably many others. Yeah, and of course, I'd like to thank you for listening. I hope you found it interesting. And of course, if you have any doubt or any questions, just ask me or feel free to send an email, wherever you want. Need to stop sharing for now. Yeah. Thank you.
Thank you.
Thanks very much. That was a brilliant talk. There was the sulfur donor at the end. I hadn't seen that before. That's very exciting. That it could work differently in humans and in fungi. Just before this meeting, Graham specifically asked me, like, is it going to be druggable? Like, aren't you just going to be helping both sides? So that that ties that up.
That's it. Yeah, exactly. That's the hope. I mean, we still obviously need to validate this, but it's the good promise that we can target them independently, block the fungus and increase the host. So that's what we are aiming to do now.
Yeah. I'm going to ask a question. Yeah.
Of course. Yeah.
So, do you think this is a process that is mostly in Canidia, and that it's mostly going to be about sort of the invasive form, or do you think it could be something? Do you know anything about sort of hyphae or sort of chronic forms?
Yeah, so we normally we test the killing capacity using Canidia. It's much easier in the lab, but we actually incubate them for quite some time. So Canidia start developing and growing. So the effect is also, counted for later stages of development. But then also, if you check so if you remember the images that I show of the hyphae growing on the cells, they do have a lower presulfidation level. So I'm certain they also are more sensitive to killing by certain mechanisms. Yeah, we haven't looked specifically, but that's a good thing we could do. For example, neutrophils are able to kill hyphae, so the long form, so we could challenge hyphae with neutrophils and see what happens. But it's certainly going to be true as well.
Yeah, that would be amazing if you could get a drug based on this mechanism that you could then like combine with, like if somebody's on azoles but they need a bit of a boost, it would be amazing.
Exactly.
It's such a different.
That's the idea, and much of the research nowadays is looking into that. So people there in NAC are looking into other ways of boosting the immune system, and that's a very promising strategy to use in combination with classical drugs. Yeah, because in the end, the immune system is the most efficient way of fighting pathogens.
Yeah. Do we know anything much about sulfur donors and whether they tend to be quite toxic, or are there any drugs that are already?
Yeah, there are quite a few which are actually in clinical trials, but not for infections at the moment. But the level of sulfide production in the lungs is being most connected with things like asthma and COPD. So indeed, it seems that it can be beneficial for both things, because there are quite a few reports already that correlate lower levels of sulfide, so lower levels of presulfidation most likely, in patients with severe COPD, and with an exacerbation periods as well. So if they provide a sulfide donor, they seem to decrease inflammation and get things a bit better. So it would be great if in that situations, in addition to get the COPD exacerbation better, we are protecting against the possible infection.
That's fantastic. Yeah.
I would think it's a candidate. If it was available as an inhalable drug, it would actually be really quite appropriate.
Yeah,
sort of the interactive surface between the fungus and the host is where you're attacking.
Yeah, exactly. So I know, there are quite a lot of studies in animals, and I know there are some things going on in humans, but the actual compounds are mostly secret at the moment, because the pharmaceutical companies want to keep the structure secret. So I don't really know if they're inhaled but most likely they're inhaled, but I don't know if in which format or anything. But yeah, we have donors that we can test in the animals, are widely known, but probably not in humans. It's just for research, but still the proof of concept, we can advance with that.
Yeah, I think one of the questions that patients would be most interested in is the potential for side effects.
Yeah.
No way of knowing that.
No, I mean, obviously it's something that will need to be carefully looked into, but it's promising that they are already looking into that for other conditions which relate, because COPD patients are of course at risk of chronic aspergillosis. So this would be a population targeting such a strategy. So there are already studies looking into this and see how they develop.
The human genetics side, the human variants. That was really interesting. So in a lot of when we have our discussions with the patients, one thing that people kind of like hearing about is like, why did I get this? And I think some people find it, the idea of being able to say, oh, I got this because I have this gene. I think they found that the idea of that quite comforting. But in the in the data that you were showing with the different, so the different types of white blood cells, you can be, it looks like you could have sort of lots of different genes that are having small effects.
Yeah.
And all like all, we often talk about sort of the immune system or white blood cells as if they're all one thing, and they're so different. I didn't realize lung macrophages live that long.
Yeah, this is extremely complex, but we as a community are learning a big deal in the last few years. So Paul Boyer, who I know is our regular speaker in these meetings, work with that actually, with genetics. So he can give you much more information than I can. But yeah, it's very interesting how so many cell types may have different effects, and so many genes are involved in that. So there are some which are very obvious. For example, the receptor on the immune cell that specifically recognizes the fungus. So of course, if that one has a difference which makes it less efficient, that has a very direct correlation, very easy to identify. But minor things in different cell populations may add up together to create a perfect storm for the development of disease. Yeah, and these are very complex, multifactorial considerations, but we are getting better and better into those.
Yeah. Okay, I think. Yeah, I know you've got a meeting coming up at 2,
Yeah,
so,
But so I just wanted to say thank you all for listening. It's been really a pleasure to talking to you. And I don't know, if anyone comes up with a question later on, don't hesitate to send me an email. Could contact me through Rayam or Beth, and I'll be more than happy of responding any question that you may have.
Can I ask you a question as a patient?
Of course, yes, sir.
I mean, obviously, new treatments are like, oh my goodness, amazing. I want it now. If this all goes through as you hope, how long are we looking at before
it's very, very difficult to estimate, and you're absolutely right that it's a very difficult, long process to get to an actual benefit. I mean, it's important to say that this is designed to be like that, to be completely sure that new things that come through are effective and are safe, because in the lab things may work very well, but then real life is different. Now, the advantage with this strategy that I presented today is that the compounds are already advanced and they are being tested. They're being testing as we speak. So the compounds to increase the host presulfidation, as I mentioned, are already in some clinical trials. So at least the safety side of it will be done. And all the other compound that I mentioned that is being developed for bacteria, that's the same. So it's being developed for bacteria, it's been already all the initial phases has been done. And I know they are now going to move into clinical trials. The first one is just to check safety. So at least we'll be one step ahead. So that could hopefully shorten the process a little bit. But in the end, we are always talking about years. Yes. Now, also on the bright side, this is my story that I told you today, but there are so many out there, different strategies. Some of them are in initial steps, some of them are much more advanced. There's, as I guess you all know, there are some drugs which are very close to be licensed to be used in clinics. So we are looking at the entire spectrum of drug development, and we hope that new strategies really get to the clinics very soon.
Okay, thanks.
You're welcome.
Okay.
All right.
Yeah, thank you so much for coming and giving us a talk. It's so nice to hear about these new treatments, and
definitely
your work's very cool.
Thank you. And again, thank you all for listening. I don't know. I hope to have the opportunity to present more work to you at some point soon. Hopefully good news.
Yeah, look forward to your updates. Definitely.
Okay, see you soon.
Yeah, thank you
Thank
very much.
Bye.
Thank you.
Bye.
Very good.
Quite a sciencey one
Yeah.
for the first
Yeah,
half. I think that's something that we are going to be moving away from a little bit, like not having so much heavy science content in the future. We've had
I think
a
it's
few.
very, although it's complicated for us mere mortals, I should imagine, especially you and Graham would be interested in it. And
Yeah, I really like his work because it's quite similar to what I did my PhD on. That was all about post-translational modifications and infections. So it's exactly my kind of thing. But yeah, it does get quite complicated.
I think he did though get the idea over.
yeah,
You know,
Yeah.
I mean, okay, it might have been in language that isn't common to us, but the fact that by increasing one thing in the host and not increasing it in the fungus, you know, and trying to get that opposite increase and decrease. I mean, that came over, and I think I've got that right.
Oh yeah, definitely.
Yeah. And I think that as an idea is absolutely brilliant. Yeah.
I think it's the
But
hallmark of good research if you can explain it in a way that anybody can understand, not just what you've done, but what the point of it is.
Yeah.
Because sometimes you see these molecular talks and you see all these complicated pictures and make all this effort to understand it, and at the end you think, yeah, but is this gonna, like, what is this for? Is it for treatments, diagnosis? Like, where are you going with this? Whereas he's kind of got the whole process right from the very, very sort of start of the research right through to and what kind of treatments might we do, which is
yes, it was very good.
refreshing.
That's good.
I think he emphasized how important the fact that we're already testing drugs for, I mean, that is a 10-year job. So you're looking at 5 years, not 10 years ahead, for if it gets through all its safety stuff. And I don't know if Beth's got a comment about this, but if you're looking at something that's post-translational and it's not terribly specific, I don't think, surely if you're just going to give an oral, just a tablet, it's going to affect every cell in the body, and the potential for toxicity must be reasonably high.
Yeah,
in
I think
ways we don't know yet.
I think it's promising that the trials that they're doing at the moment are a similar patient group. So like people with COPD, so we know it's going to be relevant, the sort of side effects and maybe drug interactions is going to be relevant to our group. I think, yeah, maybe it's just a case if you would just use a short course of it.
Or like I say, localized application.
Yeah.
Because all the issues seems to be in the macrophages in the lungs and the lung tissue, and so let's just keep the drug in there, and it might work a bit better. So it's just one of several of all the different alternative techniques being looked at now.
Yeah, I think it is. Oh, sorry, carry on.
No, I was just going to say, I was wondering, I forgot to ask him, would it be likely to be, it's too early to ask probably, a one-off treatment, or, you know, a once every couple of years treatment that would carry on working in your lungs, make your own cells active? Or would it be something you take every day?
It would. I think the effects would stop quite soon after you stop taking the tablets.
Oh, right.
But if it was something like maybe you take a course of it when you're having an exacerbation, or it would really depend on the toxicity, or maybe sort of you take it and then once the Aspergillus has stabilized a bit, you come off it. You know, like with some antifungals, like once people are sort of stabilized, there is a bit of a move towards like take the antifungals away and see if somebody still needs to take them.
I've had a micafungin recently for 3 weeks, and I think the bloods showed that I had less IgGs or IgEs or whatever, you know. So
Yeah.
hopefully, you know, that's worked. 3-week treatment, you know. So yeah, so maybe it'd be like that, and now and
Yeah.
again, sort of blast.
Yeah, but I think it's really important that like you guys are talking to those guys at this stage, like before the research gets designed, because you guys are the ones who kind of know what the disease, like how the disease plays out, and sort of when you might need a sort of a boost, a bit of a top up. Like
6 months ago.
Yeah, unfortunately, yeah.
Yeah.
I'm glad the microphone helped a bit though. Yeah.
I mean, it's great, and it's great that studies are already going on or further on for COPD, and I don't know, well, bronchiectasis, I'm still not really sure exactly what it is, but yes, for that too. Where does bronchiectasis become COPD? I'm sure you've done a talk, or somebody's done a talk on it, but it hasn't stuck in the brain.
Okay. Yeah.
Yes, they are defined according to the lung function test, which Beth talked about, I think, last time. They're quite distinct in there. Some of them trap air and some of them can't get air in and out and that type of thing. So it's a functional definition.
Yeah, so some out of people with COPD, some of them will also have bronchiectasis. Like bronchiectasis is like structural changes like that you could see on a CT scan.
Right.
Where the air vessels, they get kind of like wider and a bit floppy and fill up with grossness. Whereas COPD, sorry.
Thank you.
Yeah. Well, you're the ones that have to deal with it. Yeah. Whereas COPD is sort of the state of the whole lungs not being able to move the air back out.
Okay. Okay.
And there's different types of bronchiectasis as well. So everything gets complicated the more you try and pin it down.
I know. And you try and understand and do the best for what you've got. When you think, oh, actually, if I'm doing that, maybe it's not the best thing. I don't know. Yes. Okay. Okay.
Yeah.
But no. It's great that these treatments are hopefully going to happen, that won't be reliant on like an antifungal that is actually affecting our own, because it's our own cells that are not working properly. And so they need treating, don't they? Ideally, you know.
Yeah. Yeah, hopefully.
Good stuff.
Shall we take a 5-minute break to go and all grab some coffees?
Sounds good.
Okay, so we'll start again at about 2:05, and Lauren will be talking to us about I don't want to say morbid, but future planning and things like, sorry, I've forgotten what the word is. I can't do my words today. Graham, what's the word?
I don't know.
Sorry, I'm not always very good with my words, and I've just forgotten what the name of what I'm trying to say is. Not a will.
Well, we're looking forward to it anyway.
Yeah, that's really cute.
Sorry, I don't know
It'll
what.
be a nice surprise.
I got halfway through the sentence and I thought I've forgotten what the word is. Power of attorney. That's the yes.
Oh right, yeah.
I just need 10 seconds to remember things. That's all.
Yeah.
Okay.
My brain's fried.
But you understood the talk though.
I already had. I already knew his work because I made a video. Would you like to see the video I made of his stuff?
Yeah. Yes.
Maybe I should have shown this first because it's simplified.
Oh, good. Terms like self-sedation was totally new to me, you know. And
we actually watch presentations from the university every other week or so. So we do see them quite a lot.
Okay.
I did like his medieval
That was
Yeah.
good.
barrage things with the roof. That was amazing.
It was good. I've still got that methylation talk to do.
Oh yeah.
It's about 2 years now.
These are supposed to look like spores.
Okay, right. I'm still trying to understand the words.
Ooh, can't say that one.
They're just pretty structures. That's all.
Hmm.
Okay.
Yeah, I think I would slow it down a bit.
That's cleared it up no end.
It begs the question what the prevalence of the TT genotype is in macrophages in the
Yeah.
general population.
Yeah. I mean,
Yeah.
even if it was just screening people that were going to have chemotherapy
Yeah.
for that, that could be a really quick win. Like if you could just check, like you know, there's all like tables for who needs to get what different kind of prophylaxis. There might be something it affects in there, just to get
Yeah,
people a bit
good.
protected before they get it.
Good to know.
Prevention better than cure, sure.
I think we're in Gwyneth's kitchen. I'll have a cup of tea, please.
On the subject of physics at school,
Yep.
and understanding laws, when I was at school, we were taught all these laws, you know, Archimedes and all that. And we were also taught Joule's laws, which is, I'm reading it now off the internet, the amount of heat per second that develops in a wire carrying a current in proportion to the electric resistance of the wire. And Joule's laws is basically telling you how to measure heat and electricity. And we were taught this at school, and it went in one ear and out the other. And then about 20 years ago, I discovered that Jules lived in the same town as I did.
Makes all the
All
difference.
his life, and developed this law. And they've actually got a statue to him in Sale Park where I played football. And none of the physics teachers at school told us that at all. We had no idea. We didn't know where Jules came from or anything. If they'd have told us that, we would have been a bit more interested in physics at school.
Definitely.
We used to use his statue as a goalpost.
It's weird the way the past reaches out to touch you.
Yeah.
I know when I did biology in South Liverpool, and there's the pharmaceutical companies in that area, and the teacher related penicillin to us by referring to Fleming Way, which runs up in that part of the world. So a local reference does help you remember stuff.
I think so, yeah.
There's you know, Joseph Lister that did all, like,
Yeah.
Listerine that did all the disinfection stuff. He's buried in a little private cemetery in Stockport,
Really?
a couple of rows away from a woman that died in the house I'm moving into.
Yeah.
Just on another note, I finished that jumper from your.
Ooh,
Oh, let us see, let us see.
that is so smart.
Oh,
That's really cool.
I know it hasn't got the dermis and the epidermis and stuff on top, but I thought that would look a bit too much.
Yeah,
For that.
I like
think
how
it's
you've done
really
that.
pretty.
Yeah, with the stripes around the neck and cuffs. That looks
Yeah.
perfect.
Yeah.
Christine, can you send me some pictures, and I'll do a couple of social media posts on it, because that's really
Of
cool.
course. Yeah. Well, I think it's really pretty. I mean, it's one way of
Isn't that
looking at the
lovely?
fungus. It's been quite pretty.
It is pretty. It's really good.
And if it starts a few
So
conversations.
thank you for that. It was, I enjoyed doing it, and I wouldn't have bothered if you hadn't put the thing on for the sock. So yeah, lovely, thank you. Yeah,
Oh,
send some
we'll have to have an
pictures.
in-person meeting, so we
What
can
have
all
you-
wear our.
What have you knitted, Doug? Where's yours?
I have to wear them.
Yeah, every in-person meeting, we will expect you to be wearing the sweater.
Right, okay. I'll keep it just for that. This is my new posh sweater for when finally I go somewhere posh. Which will probably be next time we meet up.
Christine, can you hold it up again? I missed it.
Oh yeah, sorry.
Gwyneth, could we have some pictures of your socks that you made as well?
Yeah, I'm on pair 2 now because pair 1 were different. Did I send a picture?
No. Can you send us
Did I
some, and
not?
we'll do some
Well,
do some stuff on it.
Mark 2 are more similar, but they're not exactly the same. But they're near enough.
Very
In
good.
shoes, under trousers, you wouldn't notice.
Like the colours.
But I have nearly finished. So I've just got the last little bit to do. So
Yeah.
So that's the long one, is it?
Sorry?
Is that the long one?
Yes.
The picture.
So,
That's the
Yeah.
long one.
so
I
on
thought
the
of doing the COVID one. I thought that would be quite novel.
Oh,
Yeah.
On the first pair of socks, I've actually put black splodges roughly where I've got my aspergillomas.
Yeah.
Perhaps
Yeah.
this pair yet, so I haven't done that yet. But I will do it.
That looks quite Christmassy, that one.
Do you think?
Look like little holly leaves.
Christine, hold your thing up. I still haven't seen it. I think you have to speak when you hold it out. Oh my goodness.
How cool is that?
It's so good, Christine.
That's really
Can
lovely.
you see it all right?
Yeah.
It's
That is beautiful.
anatomically correct with the bifurcating hyphae.
The only problem is it's sewing in all the. Well, you have the same. It's sewing it all in at the back takes forever. Yeah. So it took me longer, not because of the knitting. Now that's done in 2 minutes while I watch the television, but the sewing up afterwards.
Does that mean no to making a few more?
I make as many as
We can raffle them off. We can have a bespoke limited run of Aspergillosis jumpers and socks, and get them raffled off and raise some money.
Yeah.
Oh, that would be nice for World Aspergillosis Day.
Yeah.
Yeah, and I've got a few pairs that are mostly finished.
Yeah, you just have to tell me what sort of size, what you think of middle size is.
Yeah. I think that's a good idea. That we can do that for WAD 2023. too. Yeah, trying to remember where we are in the calendar then. Yeah.
So, Beth, on your socks, what did you do on the foot part? Because I was running out of yellow. So I've just done various stripes and things. What did you do on yours?
I think I just did. Yeah, I just carried on in the same stripes as before.
Right.
I think.
Yes, I sort of have.
Yeah.
Yeah.
I think it looks quite nice when the different. I kind of did that for simplicity though. Like I think it looks nicer when there's kind of different types of patterns within it. But I didn't want to overload it.
I was thinking if I made another pair, I'd make them plain from the heel down.
I think I did a couple of plain ones just because I needed to take photos of all the different ones. I was like, I don't have time to do all of these. So I just kind of did one of each pair and didn't quite finish it.
Graham, I feel left out. I think we might need to get knitting something.
I did try. I started knitting when I was about 12. And I stopped knitting when I was about 12 and a half.
It gives you something to do with your hands while you're watching the television. That's why I like doing it.
That's what my phone's for. That looks like a sweater to watch tennis with, or cricket.
Or cricket, yeah. It's a bit like
cricket. I was thinking cricket. Yeah.
I was trying to be topical, you know. Wimbledon's on at the moment.
The football the other day.
I'm going to have a drink if everyone else is having a drink.
Everybody, just the whole of I don't know if it was the same way where any of you were, but the whole of Cheadle Hulme and Macclesfield, it all just went absolutely bananas. After we won the football.
Oh.
I don't watch. Hate it.
Chris doesn't watch. She does knitting.
Yeah.
I didn't watch it. I just saw the chaos afterwards.
I watch for the family.
I could just hear my husband shouting in the living room. I was just pottering about trying to watch the second, caught a bit of the second half, but that's it. I'd rather watch the cycling. So yeah.
Is that Channel 4? Is it Channel 5 now?
Football or the cycling? Which one?
Say the cycling. Is that?
No, is that with ITV? I think. ITV. We watch the highlights on ITV. It's quicker. It's quicker than watching the live coverage for hours. Yeah.
Yes, not a lot happens in cycling for long periods of time, and then lots happens all at once.
See, this is why people don't watch it, because they think not a lot happens, and loads happens if you watch stage 2. Chaos, carnage.
They arrested the girl who caused the crash, didn't they?
They did.
Oh, good.
I'm very well. The thing is, I'm a bit, because over the years there's all sorts that's happened, and don't get me wrong, I believe it was incredibly stupid, but I don't think there was any malice in it. So when they were talking about suing her, I was like, that's a bit much, because you've had cyclists pushed and you've had urine thrown on them and you've had lots of stuff and nothing's ever been pursued. And so she's just getting a €1,500 fine, which is...
They've got charges now, haven't they?
Yes, they have. They've completely, the whole thing about suing her has gone out the window, thankfully. So stupid, really stupid. But, unfortunately, we don't lack stupid people in the world. So, you're always going to get stuff like that. But she caused a hell of a lot of damage, that's for sure.
And the potential for it to be so much worse.
No. Grandparents have probably disowned her. It was their sign, wasn't it? She was waving to the grandparents.
Yeah, it wasn't even a protest, was it? It was just a friendly hello.
But to be fair, they did enough of their own falling off in that stage as well. There was the second accident after it was even worse. So it shook the standings up a bit. It's made it a bit more interesting.
Is El Diablo still running after the riders?
The clean-up bus.
Well, there was a bloke dressed up as the devil used to be in every stage. He used to, at some point, you'd see him running after the cyclists. You don't know now.
I didn't know that.
Oh yeah, I'll get a picture of him. I think
That's
he retired.
amazing.
He's probably dead now. Do you know when you get old, everything else is really, really long time ago, and it was in your lifetime. You start to feel very old.
Yeah.
I did a race a couple of before I had my little girl. So we're going back about 2.5 years ago. I did a race and they do it in Delamere Forest. It's called Hell Runner. It's a half marathon through the forest, and they have it's Hell Runner. So the devil features as their kind of the logo for it. But we had a guy on stilts walking around dressed as the devil, like at the beginning, and there's loads of like flames and smoke, and it's a big hoop. And he fell off and broke his leg. The race ended up starting late.
Oh wow!
Oh yeah!
Oh my god!
Gosh!
I'd make you cycle a bit faster, wouldn't it?
I can't believe you've never come across LDL.
No, I haven't.
No.
I'm gonna have to ask Mark now. See if he knows. He's the one who got me into the cycling.
My son does a lot of cycling, so I'll have to ask him. He watches the Tour de France and everything.
Yeah. Yeah, we used to do a lot of cycling, and then we had kids. And there's now sadly not enough hours in the day. The only cycling we do is if we do mountain biking and stuff. And we've got the youngest goes on a seat in the oldest is probably he's 5 in October, and he loves mountain biking. So we're hoping he's gonna be at the Olympics at some point.
I can't ride a bike.
Really?
I'd never had a bike. I didn't have a bike when I was little.
Oh, I'm feeling like a Phoebe from Friends episode here, where we need to get you a bike and get you riding with the little.
I have twice been on an exercise bike.
Yeah.
She fell off that.
That doesn't count.
But yeah, girls didn't have bikes. My dad said it was far too dangerous for girls. So my brother had one, but I didn't. So that's how old I am. That sort of idea was going around at the time.
Wow.
Well, I'll tell you how old I am. I can remember Eddie Merckx, and they mentioned him the other day.
Yes, because Cavendish is going. Cavendish is not
record,
saying he's going for it, but he is. He's going to go for it. He's got to go for it.
I also remember England winning the World Cup.
Bloody hell, Doug.
Yeah.
That's kind of cool though.
It is cool.
It is really.
I was listening. They were talking on Radio 4 the other day about how for a lot of the players and a lot of the younger people today, they don't, because they weren't there for 1966. They don't. The kind of hype isn't always there, and it isn't always the passion's different because they don't remember any of that. They don't. To them, it's just their little jot of life, and that's what was really interesting, actually. Yeah, it must have been quite cool actually to watch them. Hopefully, you'll get to see it again, Doug.
Yeah.
Well, that was in the days when the world was black and white.
Yeah.
Unless you're posh.
Well, unless you were posh. Yeah, I've never been classed as posh.
We were one of the many, many millions who got a colour television because my dad saw it on pot black.
Yeah.
He couldn't go back out.
Yeah. That was after 1966, though.
I was only 4.
You know, surprisingly, we got the when the Disney Channel came out, the Disney subscription service came out. We got that, and my little boy surprisingly really liked the old. They've got the old Mickey Mouse on there. There's the black and white Steamboat Willie stuff, and he loved it. So you'd think he'd be like, "Oh no, I want it in colour, mummy," but he just wasn't. He really liked it. Weird.
You should watch some of the old Felix the Cat ones, the black and white ones. They're pretty weird though.
Yeah.
I remember those ones. Well, because they used to replay them. Yeah. Anyway, shall I start? Shall I make a?
Yeah, I was going to try and do a segue into it, but I don't. Shall I try?
You can. You can. You want?
Mildly funny story. After the football this week, and the whole of Cheadle Hulme, they were just all going completely crackers, and a load of drunk men from 2 streets over started shouting, trying to ask me what I was reading, like over 2 sets of train tracks and like from the top of the building. I don't know why they were doing that, but they weren't very happy because the book I was reading, it was one of Graham's ones about talking about death. They're like, oh, that's
That's a good, that's a
they
good
didn't want to
book.
talk to me after that. So that'll do for linking.
Thank you.
So, Beth, have you got the slides? Have you got the slides there?
Yes, hang on.
So, I'm going to do a little bit of a talk. Now, you might already know a bit about this. It's about legal power of attorney. And the reason I thought I'd do a bit of a talk on it is just because in the last couple of weeks, Beth and I have both separately, with our respective families, had to deal with. Excuse me 1 second while we just move that. We've had to deal with our own family issues in terms of legal power of attorney, and yes, I thought it'd be quite a good thing to perhaps do a talk on and see if that's helpful. So just a bit of a disclaimer first. So while I've made every effort to make sure this presentation is accurate, it doesn't constitute legal advice on an individual basis. So please don't try and sue me because I haven't got much money if you do. I'd have to sell one of my bikes, and as much as I don't ride it, that'd be a bit disappointing. But so did you know that in the event of something happening where you become incapacitated, a relative can't just walk into a bank and access your pay, even if it's to pay for care, and that goes for even if you're married or in a civil partnership. So a lot of people assume that, and I know this was an assumption I kind of made as well, is that your spouse would automatically be able to deal with your bank account and pensions and make decisions about healthcare if you lose the ability to do so. But unfortunately, it's not the case at all. So thinking about what would happen if we could no longer make our own decisions, it's not a pleasant thought. I know it was quite an uncomfortable one to have with my family, but it's important to consider how much worse it would be if the situation did arise. For example, obviously if there was an accident or a stroke or a traumatic brain injury or anything like that leaves us incapacitated. So the lasting power of attorney it's a legal document that lets you, who will be classed as the donor, appoint one or more people who are then known as attorneys to help you make decisions, or to make decisions on your behalf. So it's effectively, it's a way of giving someone you trust the legal authority to make decisions on your behalf if you lose the mental capacity to do so in the future, or if you decide there's decisions that you don't want to make for yourself. And there are 2 types of lasting power of attorney. You can make one of them, or you can make both of them. It obviously depends on the circumstances at the time. There's one for financial decisions, and there's one for health and care decisions. So in terms of the LPA for health and care decisions, it's a health and welfare legal power of attorney, and that gives your attorney, so the person you appoint, the power to make decisions about your daily routine if you were in a care home, so things washing, dressing, eating, or if you were in a home as well, in your own home. Decisions about medical care, if you're going to move into a nursing home or a hospice and things like that, or if you need medical treatment. But it can only be used if you're unable to make your own decision. So if you become incapacitated, it can't, as long as you've still got capacity, it can't be used. Then there's a legal power of attorney for financial decisions, and this can be used. This one differs a little bit from the health and care one, because the health and care one, as I said, that can only be used when you use capacity. But the financial one, it can be used while you've got capacity, but it can also be held in readiness in the event that you lose capacity to make those decisions yourself. And obviously that's your decision at the time you make the appointment, if you want someone to, which way you want that to work. And an LPA for financial decisions can cover things such as if your mortgage needs paying, just paying your bills, collecting any pensions or benefits. And obviously, if it becomes necessary, they can make decisions around selling your home. So an example where that might be used is if it's to fund care for a nursing or residential home. So, Beth, if we just go on to the next slide.
Yeah.
Thank you.
In the wrong screen.
No, you're good. That's the right
Oh, no,
slide.
that's right.
So obviously, you know, you might be thinking it doesn't affect me. I don't need to think about it. You know, maybe in a few years we'll think about it. But the thing to remember with lasting power of attorney is you can only set it up when you have mental capacity. If in the event you lose capacity and then it's too late, it can make things difficult. So things like paying bills, collecting those benefits we were talking about before, or making any decisions around care. So to manage those things, your family and loved ones or whoever's kind of, you know, whoever you've got around you, they might not necessarily be able to make those decisions. So they'd need to apply to the Court of Protection to become a deputy. Now that can be a very costly and long drawn out process while the court weighs everything up in order to appoint a deputy. Just next slide for us, please, Beth. So obviously, because this all hinges on mental capacity, obviously just going to mention a little bit about what mental capacity is. So I know, Christine, you'll probably know quite a bit about this from your background, but so obviously just to go over it, the Mental Capacity Act 2005 says that a person is unable to make a decision if they can't do one of the following. So that's understand information relevant to a decision, retain that information long enough to make the decision, use or weigh that information, or communicate that decision. So some people will be able to make decisions about some things, but not about others. So you might be able to decide whether what to buy for dinner, but not know how to make or arrange home insurance. And obviously, in cases sometimes with dementia and Alzheimer's in particular, there can be days where they're quite lucid and you can make decisions, and then the next day it might not be quite that lucid, so it can change from day to day. So if someone needs time to understand or communicate what they want, it doesn't mean they lack any mental capacity. So for example, dementia doesn't necessarily mean that someone's unable to make decisions for themselves. And where someone is having difficulty making a decision, obviously an attempt will always be made to try and overcome those difficulties before it's just kind of slapped to it. You lack capacity. And then to help that person decide for themselves. So when you make an application for lasting power of attorney in England and Wales, a certificate provider who has to decide if you're capable of making that choice. Obviously, while you're filling it in, and you're supposed to still have capacity. So it can be someone who's in a professional capacity, has known you for 2 or more years. So that could be a GP, a lawyer, or a social worker. It's a little bit different in Scotland, but none of you are in Scotland, so that's okay. So if you wanted to make an application for a lasting power of attorney, you do it to the Beth, do you want to just switch slides again for us, please?
I'm trying to. I think I'm in the. Oh, there we go.
So I feel like I'm on one of Boris Johnson's talk where Chris Whitty's saying, next slide, please. So in order to make an application for a lasting power of attorney, you would contact the Office of the Public Guardian, and they would be able to send you the relevant forms either digitally or in the post. And you can also get an information pack from them. You can fill out the forms yourself, or you can ask a solicitor, instruct the solicitor to assist you if you're not sure of the process, or obviously if you've got quite complicated financial affairs and you're not sure how to go about it. In order for a lasting power of attorney to be in place, it has to then be registered with the Office of the Public Guardian. So that's where you send the paperwork back to them. It takes about 8 or 9 weeks to register it. There is a fee. It's £82, but can be reduced if you're on a low income by 50%. And then it takes about 9 weeks. If you lose capacity in those 9 weeks while it's being processed, it can still be registered. So it's kind of, it can be done that way, but it can't be made once you lose capacity. There is a difference between a lasting power of attorney and an ordinary power of attorney. So an ordinary power of attorney, that covers decisions kind of in an interim period about financial affairs, and it's only valid while you have mental capacity. So it might be where you just need temporary assistance. So if you're in hospital and you're confined to the house and you can't get out and you just need someone to act for you, that's a bit different than how you apply for it. You would go to Citizens Advice and they can give you there's a kind of legal template for how that should be completed. Now there's loads of places where you can get more information on lasting power of attorneys. There's Age UK is really good. Citizens Advice, the NHS website's got some on there, gov.uk, and yeah, that's it, brief but short. But I just thought, as I said, it's I know it can be a difficult thing to talk about, but it's something Beth and I are, reasonably, youngish years have had to deal with our own families, and it's something that does make you think, oh, this could be something that's of use to other people, because my family certainly didn't realise that they're stuck in a position at the moment where they don't have LPA for my uncle, and they're now in a bit of a battle with some doctors about his care. So it's certainly highlighted the importance of just thinking about it. And I actually might be getting my husband to make one on my behalf. I'm applying to be an on-call firefighter, and it's actually one of the professions where if I'm successful in doing it, I'm not leaving it, it's just on call, you're retained. It's one of the professions where they actually recommend that you have an LPA in place in case of an incident. So it's I thought it was quite a topical thing to think about, and not raising it in any way because I think any of you are going to go off and lose capacity anytime soon, but I just think it's something that's certainly worth thinking about. And
and it's not even a huge amount of hassle to set up. So I've actually got one here that I'm signing for my parents.
It's like Blue Peter. Here's one we made earlier.
It's not a huge document at all. It's just a few bits of paper. And once it's done, you can just put it on one side and just forget about it for years and years and years.
I had a lasting power of attorney, the financial one for my mum.
Yeah.
And we actually went to a solicitor who made the judgment that she was capacitated enough to be able to sign it and say yes, that's okay.
Yeah.
And it was sent off, and it was an immediate thing that we had.
Mm-hmm.
What I don't know is if you fill it in and it's lodged there ready, and then you have an accident that incapacitates you to the point where you can't say yes, you can activate that now. How does it become active? Is it just that the person that you've named goes and says, my mum's been knocked on the head and can't do anything?
So for example, if it were in the event of the health and care lasting power of attorney. So say for example, that's only in place. So if my husband had one for me now, he can't turn around and go, oh, I've got an LPA, you can't have that operation because I've still got capacity.
Yeah.
As soon as I am deemed to not have that capacity anymore, he then would step in and he would have a document and he would say to any kind of care providers, I am lasting power of attorney, I can make these decisions, and he would then make those decisions. With the financial one, in terms of the financial documentation, what you would have to decide and what would be written into that documentation beforehand is at what point they're taking over of your finances becomes in place. So are they going to help while you've still got capacity, or is it going to come into place afterwards? So again, it would be a case of when you go to the bank with the documentation, if it were the bank or to collect all the pensions and credit service, you would have that certificate that would say they've now been deemed to not have capacity. Obviously, you would then have some form of documentation around around capacity from a medical point of view.
Right.
And then you would present
so you would have
two
the
things
form,
together.
Yeah, so you'd have the form and something from a doctor or the hospital to say you had dementia or whatever it happened.
Yeah. So obviously, in those circumstances, then a mental capacity act assessment would probably be undertaken by the medical team at the time. And I know, I don't know. Used to know a bit more, but I've been out for a while. But then obviously you'd probably go down the whole DOLS, deprivation of liberty standards and things like that would come into place. But you'd have some sort of documentation in place from a nurse or clinician point of view.
Yeah, from a professional. Yeah.
Well, using that example you just talked about of going into hospital for an operation.
Yeah.
And you would have capacity, but as soon as you're unconscious, are we talking about capacity or are we talking about something else?
No, it's well, I suppose you suppose you're temporarily incapacitated, aren't you? But you haven't been deemed to lose capacity while you're under
so you can't get it done while somebody's just asleep then.
If somebody had to make a decision, that would go to a next of kin, so that must be a distinct entity.
Well, believe it or not, it's a really grey. This is where this is because of my uncle is where it's become kind of. I've realised just what a grey area it is. It's just people. There are a lot of assumptions that because someone is a next of kin that they can make those decisions, but actually, if you get a medical team who disagree with your decision as a completely as a next kin, they don't think it's in the best interests of the patient. So the example is with my uncle at the moment is he's got a history of schizophrenia and drug abuse, and there's a lot of mental health issues there. But he's now been diagnosed with terminal cancer, and 2-3 weeks ago he was deemed to have capacity, and we were just gonna. My nan was going to bring him home and care for him with my mum and stuff, and give him those last, because that was his wishes. He doesn't want to be stuck in hospital waiting to get COVID and die of that instead of the cancer. And now they're deeming him to not have capacity and won't let him come home, and they want him to die in hospital. And my family want to bring him home, and they're kind of locked in a little bit of a battle at the moment about his wishes and what was best for him. And it's proven incredibly difficult, because the last thing the family want is, because of COVID as well, COVID is just not, what you want to take out at the moment, but you can't, you make assumptions as of next of kin about what you'd be able to do, but there's so much more to it, and it's just a huge it's even down to things like, so if you become, so say if you develop a brain injury and then you need to go into a home for care because you can't be looked after at home. If you don't have something like this necessarily in place, it might be that a social worker decides that they're going to make decisions with the medical team because they believe that that's in the best interests of the patient, and you can't really get a say over it then. So it's a grey area with the health one, because you might get a medical team who just goes, yes, you are next to kin, and you said that you know what the wishes are. But we've all seen in the news over the years, where particularly where children have been concerned, where actually the parents haven't been able to make any of those decisions. And it still doesn't mean you'll be able to make certain decisions, but it's great. I think it's a bit more clear cut with the financial side of things, because it gives you the ability to manage the finances and collect benefits without much kind of hindrance or legal, jumping through legal hoops, because you have got the permission that was granted before capacity was lost to do that. But it's
I found once the financial power of attorney was enacted, the people I had to give, the banks and utilities and all sorts of people, they were so good. They were so very helpful. There was no, what do you mean? What's all this? Once they'd seen a copy, they bend over backwards to help you. So, you know, it wasn't a scary thing once it was enacted.
No, and you just think from your point of view as a relative, Christine, that must have just made life so much easier, because you weren't, because I mean, having to go from, I've read some stories about going through the court of protection, and it's horrific. It's drawn out, and a judge will assess the circumstances, and you just think, God, that's in light of, say, potentially an accident that leads to reduced capacity. You just think, God, is that what a family would want on top of it, is having to argue the case? It's just.
What about in the case of a living will? Because I believe in a living will, you can say you wanted to die at home, or.
Do you know what? It's not actually something I've looked at a living will from some obscure memory that's in the back of my mind. My thinking is that a living will is more to do with that, and you know, whether you want to be on a ventilator and if you want treatment withdrawn. But I can certainly have a look and find some more stuff and pop it onto to Facebook, because I put all these links up as well. I said it's not, and I understand it's not a conversation that a lot of people want to have around these things, but I think, I've got a really good friend of mine who lost his wife 2 years ago in a really tragic accident, and they didn't have a will, they didn't have insurance, they didn't have anything, and he had quite a bit of a mess. She was 35 at the time, and he had 2 children, and he had quite a bit of a lot of stuff to deal with that he probably could have done with. But you don't want to think about it. No one wants to think about it, and sometimes things just kind of, maybe I should. Maybe I need to do some adulting and think about it.
Yeah. Well, I went to Age Concern. My mum had dementia, and Age Concern had a series of classes, if you like, on how to treat people with dementia and all the things around it, and power of attorney and all that sort of thing. And there was one lady there whose husband had early onset dementia, and was okay for a lot of the time, but now and again, he really confused. And they went to the bank and they were getting some money out, and she just happened to say to the teller, "Oh yes, well, he's having a good day today. He's got dementia now." And that was it. They froze their bank account. It was a joint account, and they froze the account.
Wow.
Yeah, and then because he would have access to it as a joint account, and if he had dementia with access to it, he could, as far as the bank was concerned, start just
flittering it away,
giving money away. And it took her ages to get access to her money. It was awful.
I mean, you can't even imagine. I mean, from what I've actually read around the Mental Capacity Act and dementia, that that's, like I briefly said earlier, from my kind of understanding around reading about it for this, is that you can't automatically deem someone to lack capacity because they've got dementia, because anyone who's ever been around someone with dementia will understand that it's not that straightforward. Perhaps in the end stages of dementia, it certainly is, but in the beginning, the people can have exactly that. They can have good days and they can have bad days. If they've taken their medications late, it can change.
Yeah. But it was just that sort of throwaway off the cuff remark to the teller, and that was it. So careful what you say in the bank. Don't make jokes.
Like at passport control, don't make any jokes. Just get through.
Just make eye contact and walk on. It's fine.
Just take the money.
Yeah, but I'll post the links up and some of the bits I used to research up on Facebook, and I'll have a look at the living will stuff as well, because there was I'll post some information up as well, because other people might have heard as well of enduring power of attorney, which was the older version, which was before October 2007, but it was replaced by lasting power of attorney. Now anyone who has an enduring power of attorney in place from before 2007, it's still valid, but you just can't make those ones anymore. It's just the lasting power of attorneys.
Wasn't the enduring power where financial and social care came together, didn't it, under one certificate? Yeah,
it was all.
Yeah.
It was
and
all
then they
together.
split it up. Yeah.
And then there's another bit of information as well. I didn't look. I'll post the links up on Facebook. But then there's another bit around people who've made applications for lasting power of attorney. They may actually be entitled to get some money back on it. So I think there might have been a few errors in calculations being made. So that might be useful for anyone who's already made one and might be eligible to recoup a little bit of the costs on it. Maybe that they weren't assessed on their kind of financial situation. Because I mean, £82, it can be something for some people and nothing, for others. But if you're making it, if you've got, because you can't have a joint application, you've got to make it on an individual. It's got to be on an individual basis, so it could get expensive. So I'm gonna have to start splitting my family up with my 18-year-old brother. He's gonna have to start taking responsibility for some of them.
That's the thing, isn't it? Who do you give the lasting power to?
I said I don't want it. I have enough responsibility with my family. They can all sort themselves out, but they spend too long killing each other to do bloody families.
There's always one person that ends up in the family having to deal with everything. Like my poor mum, always, whatever weird messes people have made, it's always her that has to go and sort it out. So like when I had an operation a few weeks ago, it was just my gallbladder, totally routine, but you never know. So I thought, like, if something did happen, I just made an envelope, wrote down what my wishes are, and said, if the worst happens, open this envelope, and if it doesn't, then just put it in a drawer or something. And I think that it's really important, once you've made your power of attorney, to keep talking to people so that they know what your wishes are for different circumstances, because that it is quite awkward to bring it up, because not just for yourself, but you think, is this going to really upset them? Like, are they going to get really funny about it? And but I think
she's after me money.
Yeah, well, no, it is, it's.
She's trying to put me in a home.
Yeah, the last thing you want to. Well, I mean, some people would, unfortunately, that would be the goal of some people, because as we said, there are some, undesirable people in there. But I think most people's intentions is to protect and look after your loved ones, but there is also, I'm not going to lie, there's also a selfish reason for it, because I'm thinking, if anything happens, I'm next of kin for my mum, and then I'm next of kin for my dad, and I'm next of kin for my brother, and I think, well, gee, if anything happens, I just want to make it a little bit easier to be able to manage those things, because we've all got, I've got my own children and my own family, and while I want to care for them and look after them, I want it, if I've got to do it, I want it to be easier,
And just like having a list of like what accounts are there, who have you got this insurance with, just having that written down somewhere will make it a lot easier.
Every time my in-laws go away, I mean, they only have to go to Cornwall, and they instruct us exactly where everything is. All the account logins are here, the deeds to the house are there, the insurance policies are here. It's like,
good.
Drive safe, you're going to Cornwall.
Yeah, and also I think sometimes people, if you don't talk about stuff, people might think they know what the other person would want in that event and be getting it completely wrong. So like once I'd come out of the operation and I was obviously still alive, me and my parents, we did have a little bit of a chat about it, and poor things, they thought that basically if anything happened to me, they would be landed with 3 piddly old gross cats, They were absolutely dreading it. That wasn't my wishes at all. And when I said, no, you do not have to take the cats if I die, just my dad, he just went, oh, thank goodness.
That was more of a worry than you not coming out of the operation.
It is, they're disgusting cats. They've always, one of them peed on me in the night last night, and that's not even unusual. They're sick in people's shoes, they're whingy, and I think the thought of having the three of them to deal with on top of like grief and everything else, I think they just did not want. But yeah,
I have to ask, Beth, what was your wishes for your cats? Who are you going to land them on?
One in the box with her.
Claude, yes. No, that was my wishes for Claude, because he's really old. He's got definitely 1, possibly 3 kinds of cancer.
Oh God.
He's old, and he's had his life. But yeah, there's no.
Hey, I can remember the World Cup.
He's old and had his life and all.
Oh no, I think you've got a lot more life in you than. Where is Claude? I can show you him. This is Claude.
You don't look old enough to remember the World Cup.
Yeah.
It's a cat.
Well, because there's more life in us, I think we're running out of time, but I'm not going to let you get away with your routine.
We tried.
For everybody. I've heard a few whinges already, but we're going to ignore them.
We've got to go easy with Doug now. We know how old he is.
Yeah. So now
I was hoping to filibuster
a
this
little
to
bit
the
of
end.
therapy and get back in the zone, and then you'll be chilled tonight. Whatever comes your way, you'll be completely chilled. Okay, so you know some of the routine. I always like to throw in a couple of new things just to keep you on your toes. Make sure you're comfortable and safe. Yeah. And you've got space that you're not going to knock anybody out. Unless you want to, of course, that's a different matter. Okay. Nice deep breath now. Make sure you draw it up right through your lungs, in through your nose, out through your mouth.
Oh, somebody's gone.
Oh, we've had enough of that. Right, hang on, folks. It's not for long. Okay, so enjoy that nice deep breath. Okay, as we always do, start. Keep your eyes open. Take your head, your chin to your chest. Stretch the neck muscles at the back. Really loosen them up. Core neck muscles don't get much, and they hold this heavy head of yours. Now to the back, as far back as is comfortable. Don't overstretch, just do what's comfortable for you. Okay, chin to chest again. And back up to the ceiling. Okay. Now to the left, again, stretch right so it gets to these shoulder muscles, includes them as well. And to the right, left again, and right. If it's comfortable for you to do so, make some circular movements. As I said to you before, I actually hate this one, but I do it because it strengthens these neck muscles. But if you hear the noises that come out of my neck, you wonder why I was doing it. And when you're satisfied, rotate again the other way. Some people love this, I must admit. I like the other one, but I don't like the circular one, because all I can hear is the crackling of my neck.
It's a neck mixer, that's the same. That's what it reminds
Well, there
me.
you go. Okay, so once you're ready now, comfortably, do some shoulder rolls in the reverse, so backwards. You can either put your hands on your shoulders if it's more comfortable, or you can just do it freely, whatever works for you. Rotate them round. Again, this is good when you're carrying things, you get those shoulders, because you feel that in your neck as well if you don't loosen these muscles up. And forwards. Right, wonderful. Now, arms out to the front, as I always do, like to get these arms stretching up to the ceiling, out to the side if you can, back up to the top. Please don't knock anyone out, and out in front of you again. Now, here's the new one for you. We're going to go, we're just going to rock our hands like this, yeah? Let's do that a few times. Now, bring your hands in, and you're going to be a boxer. So rotate your hands like that, forwards, that's it. And then stop and go inwards. Okay, wonderful. Hands down. Just loosen your shoulders again now to relax them again. Lovely. Now. Know what's coming next now, don't you? So bring your knee up as far as you can. Just grab that knee. It's moaning already. Got patients online and they're not moaning. What are you doing? And the left knee. Bring it up to your chest as far as you can, if you can. Only what's comfortable. And now feet to the floor. Okay. As we do, right foot, rotate your ankle on the ball of it. So you're on the ball of your foot and you're rotating your ankle outwards. Yeah. What did she say? Ball of your foot. Ball of your foot, yeah. So you're resting on the ball of your foot and you're rotating your ankle round because you get full range of movement if you do that. Yeah.
You can't do that.
Can't do that. All right then.
No, you can't.
Inwards, kind of loosen your ankle muscles, you see, so that it all strengthens it. Voids increase this. Okay. Now on your left foot, ball of your foot, rotate your ankles round. Yeah. You can't do that's your homework, Doug. That's your homework now. You see, practice that. And inward.
It's not the physical bit, it's the mental bit.
It's really
coordination.
If it makes you feel any better, Doug, I'm struggling a little bit with the coordination of it as well.
So let's have another deep breath.
If you stood up.
The last one, because I know someone's going to need the energy for this one. Okay, deep breath again. And out. Right, now just lift your feet slightly off the floor and feet forwards and backwards in turn. So your toes to the floor and up, toes to the floor and up. So you're just pulling your other ankle muscles now forward and back, yeah. And then you're on your imaginary bike again. So put your hands on your seat so you've got something to support you. And then just lift your legs slightly because this is working them abdominal muscles. And now we're on our little bike and we're pedaling. So keep pedaling, yeah. If it gets tiring, put your feet down on the floor for a minute and stop. And then just lift them again, the down. And this is a clever bike, this, because it goes in reverse. So turn your legs reverse. And then down on the floor. And then tonight when you go, oh God, my stomach's killing me. You know that.
I'm talking to somebody who can't ride a bike. We've already said that. Yeah. Pedaling backwards is hard.
Okay, nice deep breath now. Clear your head. Find your place. And remember, as I always say, yeah, hands out, hands to shoulders, and give yourself a hug. And remember, be nice to yourself and give yourself time and space, 5 minutes in a room, just to chill, ride your imaginary bike, whatever it is you want to do, you know, balance on the ball of your foot. It's all good, but just enjoy. Okay, thank you very much.
Thank you.
Thank you. Thanks, Chris.
Bye-bye. Don't come out loud.
Yeah. you were just thinking
you love it, really. Don't lie. You love it, really.
Graham, before everyone leaves, are we gonna ask us all about future meetings, what they've already done that over the beginning?
Sorry, I can't tell what you're saying.
Make any of that out.
Oh, just a quick. No, I think we'll do it next time.
Okay.
But we're going to reassess. In fact, probably we could reassess all of the meetings that we do, Thursdays and Fridays, and see if anyone can think of. We want it to be what you want for these meetings, rather than us speaking to you so much and telling you what we think to do. Except the exercise is compulsory, is that right?
Yeah, there's no escaping exercise.
So have you got any ideas that we could do if if a patient wanted to give a talk, that type of thing, or a carer, or do you want us to do we have the capacity now, because we'll still be on Zoom for a bit, probably till sort of end of the year, I should think. We can split meetings up into groups. So, for example, I remember us splitting up carers and patients at one time into 2 separate groups, and that seemed to go quite well. But that was in a face-to-face meeting, and we can actually do that on this now. Or maybe you'd want to, maybe I would go into one room and Beth would go into another room, and so on, and maybe we could do 2 rooms or 4 rooms, and you could choose who you want to go and have a chat to, that if you want to one-to-one. One of the things I thought might help, I might start offering, or we might start offering, is a one-to-one meeting with a member of the team at a particular time in the week. Might be good, because I think some people might not like the big group, and they might want to be more of a one-to-one or a couple of people to one type situation. So it's just a blank sheet of paper. We were starting to come out of COVID now. we have the capability of coming back face to face in a few months. Do we want to carry on doing it this way? I think we need to do a hybrid meeting and try and do both, because a lot of people it's quite convenient doing it on Zoom, and we have the capability to do both as well. I would throw the doors open and have a big discussion. We were going to do it today, but we've run out of time, so we'll have to do it next month, or maybe the month after, because August is quite a quiet month. So that's the plan anyway. So I'll put a lot of this into the Facebook groups as well. Interestingly, the people we want to try and, one of the groups we want to try and get to are the people who don't come online now. And most of them can't come online, so we're never going to be able to reach them, but it might be worth asking them some questions about what they would want out of the group as well. So we'll have a go at that next month or the month after.
How we build back better.
Indeed. That's a catchy phrase. No, someone should use that.
One of the topics I'm interested in, but I probably may not understand it, is exactly how the drugs work. So for instance, I've been on Seretide for ages, but recently been given another bronchodilator. And I'm interested to know how they both work, how they work at the cell level in a simplified version.
Yeah, it's getting it simplified, but not so that it's useless. It's difficult, isn't it? Yeah.
I know it opens up the airway, but I'd like a bit more than that.
Okay. Yeah. That's
and what happens if you're an hour late taking it, and what happens if you take it half an hour too soon because you forgot you didn't take it that time yesterday? Just those little, it's just of interest.
Yeah. Do you know so we're getting our new clinical fellows in next month, and in their induction, one of their talks is from I think it's one of the nurses that looks at all the different asthma treatments. When I see her, she'd probably be just the person to do this.
Yeah. Okay.
She'll be able to tell you.
Because the thing about it, the technique, someone needs to talk about techniques of using these things, because often when we went to one of the meetings some years ago, one of the things we discovered was that a lot of people, they were not using things with, it's the wrong technique, and so therefore they weren't getting it into the system because they weren't using it properly. So that's another one that, not saying you are going to do it, but there is some people who just you know, they all vary so much. So it's just knowing that they're using it correctly, and instructions to do that is helpful, I think.
Yeah.
What I would comment on that is my new inhaler, which is a Braltus. The instructions that come with it are pretty clear. You know, breathe in till you hear a noise or whatever. Hold your breath for 10 seconds.
Some people believe that following that instruction that they are doing it right, but actually when they're observed by somebody, you know, like a physio or a doctor, they actually find that they're not quite doing it, you know, and then it's important drug by not having that proper technique, and that I think sometimes it is difficult to be observed.
I was just going to comment that with the Seretide, and I've got a spacer, I have been checked ages ago doing it, but there are no, I personally, maybe I've thrown them out, I've got no instructions on that.
Oh, Gwyneth, just on that one, I did post a link into Facebook the other day. It was from the British Lung Foundation. They created a load of, I'll re-find it and I'll send you a messenger. So they
covered, that's fine, yeah.
Oh yeah, but I think what you're saying, because so the British Lung Foundation, they, with the Task Force for Lung Health, they produced so for each inhaler they've got a load of videos that goes through which one. But I think what you're saying, Chris, about people actually observing technique, because it's like, well, it's like when you go to the gym, you know, you can watch instructions on how to do a particular move, but until someone watches you doing it and goes, oh, actually, no, that's a bit wrong. So yeah, but I'll find the link again, Gwyneth, and I'll send you it on.
Gwyneth, maybe. Oh, sorry.
Sorry, we're having an argument.
Oh, I was just going to be flippant and say, Gwyneth, you take your inhaler on the Zoom call, and we'll all tell you what you're doing wrong. It'll be fun. Oh, I can't imagine anything worse. Yeah.
Thank you for a brilliant meeting. I really enjoyed that. Yeah, Doug's gone off for a rest.
Chris has finished him off with an exercise.
Got to lie down.
Yeah. The power of attorney thing is important, and it's something I know I should do, but I haven't done.
Thing is, it's like all things like this, isn't it? It's like I haven't updated my will for years. You know, since I was 20 and I went in the army. You know, I've never updated it, and now I've got 2 kids and probably should update it. You know, so
you should do.
Yeah.
My children were young when we did ours.
Yeah.
So it's probably.
You have one, but
we should do.
Even just down to things like I've got, you know, we've got a few life insurance policies between us, but God help anyone who has to find them, because I know where they are. But so it's just things like, yeah, before we drive to Cornwall in the morning, I'll be putting all the documents in one place just in case.
Okay.
Right, Gwyneth. Say that again, sorry, Gwyneth.
Have a lovely holiday, Lauren.
Thank you very much. Take care. Have a lovely weekend.
Thank you.
Bye.
Bye.
Bye. Thank you, everybody.
Graham, have you got 5 minutes? Oh, sorry.
Okay.
Is that the wrong?
I'm struggling to
right. Carry on.
With the Learning Hub stuff, the stuff that's already on the Learning Hub portal, would it mess up your system if I transfer that onto Teams?
No.
Cool. Because I was thinking, like, there's the general, you know, the ID NAC channel where we do the meetings.
Yeah.
There's nothing in the wiki in there already, so that might be.
So that I think that's good. I think it's good to change. I think it's a better place for it. And if you are leaving it to you, if you think it's a better place for that information, then I think you should go for it.
Okay, cool. Because I liked the way you done it, but I don't know whether people are looking at it.
I have no idea. So I think if we're addressing, if it's internal information, putting it under the internal system that we're all accessing is a good idea.
Cool. The other question I was going to ask is, apart from, so there's the ID NAC general team.
Yeah.
And there's the other one for the joint MDT with micro is a separate team. Do you know of any other teams that are floating around? Have you come across any? Just to make sure that I'm not missing any.
Not general ones. I don't think, I wonder if the juniors have got one. I suspect not.
Oh, maybe, yeah. Because what I was thinking, it would be really nice to have, so like the clinical fellows one is sort of more like induction focused and for information targeted at them. But then other groups might want one as well, like the lab might want one for their inductions.
Yeah, that would be cool.
So there's not really a cap on how many we can.
No, that would be great. I wonder if the lab's got one, got a team. I would presume they must have.
Yeah, I don't know, because they do a lot of in-person meetings. They just grab everyone.
Yeah.
Yeah, maybe I'll ask Caroline. Yeah.
Yeah, good stuff. How are you feeling? Yeah, you've been struggling a bit this week.
Absolutely fried.
Yeah.
Just revolt. Just, oh my goodness, no. But I've got a list of things to try that should help.
Okay, cool.
Yeah, so things like some people don't take it every day.
Right.
And I think that could possibly, because it kind of seemed to build up, and then like after 5 days in a row, it's like climbing the walls.
It's going to take a bit of time in a process to get it right, isn't it?
It's learning how to have a new brain, because it doesn't work the same way as before.
That's in the old brain. You're all right.
Yeah, so like I have like done my full days this week, just at really weird hours, really strange hours.
Well, you're doing quite a sort of what's the word? Sort of enclosed project that you can just concentrate on, and I think that's quite a good way for you to work, especially at the moment.
Yeah, but also those sorts of tasks are the ones that I get completely locked into.
Yeah, I noticed. Yeah.
Just like, and I'll be sat there saying, I need to eat now. I need to stop this, go and eat, and
yeah, I think
won't comply.
I will bug you every day, because we've got to make sure you're all right, because you're on your own. We just want to go, hi, everything okay? And yeah, I'm fine. Other than that, I think that's fine.
Okay, good. I'm just aware it's a weird. I don't want to cause too much disruption.
Well, I did my appraisal in the week, and basically it seems,
how was it?
It just comes down to me. So I asked about how do I work the ESR system on absences and all this sort of stuff for people's holidays. And they said, oh, we don't use that. We've no idea how to use that. Okay, so if Deputy Directorate aren't using it,
Yeah.
The systems are there and they don't bother using them.
What
so I think
do?
we just need to keep a grip on it and be aiming to come back, more than 1 day a week, then more than 2 days a week, and so on, a bit like we did last time, as you get on top of it. But you're doing plenty of work at home. It's not an issue really when it's not time sensitive work, is it? So the thing was, is there a newsletter due?
Yes, it's on the verge of being sent out.
Okay. I think it's the time sensitive things. It's this meeting, which I think I might start putting a little reminder in for myself to remind everyone about, because we forgot about it this week, and maybe the newsletter thing, just to make sure that goes out at a regular time.
Yeah.
And I think the team newsletter is something we should start, but I can do that if it's easier that way. Just to they may not even read it, but if we're sending them something every week, new people are going to be able to, oh, is that there's a team up there and that's what they do, sort of thing. So that's the the aim in doing that, at least initially.
Yeah, I've started putting a template together for that, but because at the moment the limiting factor is populating it with stuff that's happening around the department.
Yeah.
But like next week, I'm going to be having meetings with people about like the induction and learning stuff.
Yes, yes,
So
right.
I'm going to.
And you can have them on. You can do them on Teams if you're not managing to get in. So that will be cool.
Yeah, so I'll just try and be kind of asking people individually.
Yeah.
Or like I think the lab and Katie from the lab both got put forward for an award, but I don't know if they won it or not.
I don't know.
So that could be something good.
And all the fellows are gone by the end of the month, aren't they? So we need to
Yes.
capture them in the next few weeks. Okay.
Yeah. I think what I'm going to do is I'm going to show Ali one-to-one how I've set things up, because she was the one that wrote the induction document.
Right.
And then if she thinks the general approach is good, which I think it is, then I'll bring everybody else in.
Okay.
Just
sounds like a good project.
I think there can be a huge amount of it. It won't run out.
Yeah.
There's always more.
And that's a really useful aspect of what we have to do. So that, you know, even if those tasks are what you achieve in the average month, and that's cool.
Yeah, and I'll talk to Rena about the antifungal stewardship module. I've started putting that on Teams, and there's a list of stuff I need from her.
Yeah. Okay. Cool. Sounds good.
Yeah. Cool.
That was a good meeting. Everyone seemed to enjoy it, so that's cool.
Yeah, I'm glad the talking about the sort of darker stuff didn't upset people.
No, it doesn't.
You never quite know.
Yeah.
Yeah.
Yes, we'll try and get more people in next time. But July, August seem to be very slow. Possibly July is the worst for some reason.
Yeah. Yeah. Summer. Yes, and I'll work out who that. I've got the name of the person that does the asthma treatments. I'll contact her about the meeting as well.
Yeah. Cool.
That should be a good one.
As long as you're feeling on top of things, that's okay.
I think so, yeah. Yeah, just need to get the newsletter sent out.
Great. Cool.
Thanks for listening.
I'll be in touch.
Yes, and so will I.
Okay, have a good weekend.
And you too.
Cheers, Beth.